Hello, and thank you for joining us today. Today's webinar is brought to you by Waters, the science of what's possible. My name is Kieron Faherty, and I will be your host and presenter for today's event. This edition of our Meet the Expert series will feature part one of a three-part series focusing on the fundamentals of mass spectrometry. Part one today will focus on ion sources. Part two will focus on mass analyzers and detectors and will be held on September 19th at 1 p.m. Eastern, 10 a.m. Pacific. Part 3 will focus on advanced topics in mass spectrometry and will be held on September 26th at 1 p.m. Eastern, 10 a.m. Pacific. First, I would like to provide a few friendly reminders before we begin today's presentation. We will have a live text chat during today's webinar, and I will be handling that text chat. We encourage you to submit questions via the text chat during this presentation. Some of your questions will be discussed live at the end of today's webinar as time permits. Questions which cannot be answered today will be addressed and someone will email you with a response shortly after today's session. Following today's webinar, we will be providing the following to you via email: a link to the recorded version of this webinar, a PDF copy of today's slides, and some additional related reference materials. As I said earlier, my name is Kieron Faherty, and I am the America's Separations and Mass Spectrometry Field Marketing Manager at Waters. I joined Waters after receiving my PhD in chemistry from the University of Massachusetts. I spent 17 years in the field before moving to my current role, most recently as a field applications chemist, working with biopharmaceutical and proteomics customers on time-of-flight mass spectrometry systems. I'd like to welcome you to part one of this three-part webinar series covering the fundamentals of mass spectrometry. The goal of this series is to provide you with some background on mass spectrometry and to help you become familiar with the systems and technologies available today. In today's session, we will look at ion sources that are commonly used in mass spectrometry. Future sessions will cover mass analyzers and some advanced topics in mass spectrometry. We will start with a review of who uses mass spectrometry in analytical laboratories. We'll look at some definitions of mass. We'll then discuss a number of ionization techniques in use today, and we'll conclude at a look at how ion sources are designed. Mass spectrometry is a very flexible and versatile technique that can be used for different analytical challenges across many different industries and application areas, such as pharmaceutical analysis, food and environmental testing, chemical materials, medical devices, health sciences, and academic research, to name a few. When deciding whether to employ mass spectrometry as part of your analytical toolkit, it is important to be aware of the regulations in individual industries, as the analytical techniques or methods required may be stated within those regulations. Mass spectrometry has become an important analytical tool to support both quantitative and qualitative analyses. It can be used to confirm expected compounds or identify unknown compounds in many types of samples. It can be employed in synthesis to check what is put into a reaction, how that reaction is proceeding, characterize the resulting material, and check its purity. As an example, in the pharmaceutical industry, it can be used to look at the life cycle of a drug in test subjects to ensure its safety. Mass spectrometry is an information-rich detection method, and there are many advantages of adding it to existing methods or assays. For example, mass detection can be coupled with other detectors, allowing users to gain more information from the same sample in the same injection. Mass spectrometry is highly sensitive, having better or lower detection limits than other detection techniques. This allows very low abundance species to be detected and analyzed. It is highly selective. Mass analyzers allow users to acquire or extract information on individual compounds from complex mixtures. And finally, mass spectrometry can be used to measure any compound that ionizes. This allows the detection of species with no UV chromophore or those that have low UV response. But before we go on, let's define mass spectrometry. Essentially, mass spectrometry is a technique for weighing molecules. Obviously, we can't do this with a conventional balance, so we use an analytical technique that ionizes chemical species, measures their mass-to-charge ratio, and displays this information as a mass spectrum. An example of a mass spectrum is shown here. The y-axis is intensity, and the x-axis is mass-to-charge ratio. A mass spectrometer detects charged species, not neutral ones. So if a species is not ionized, an MS system will not detect it. As we said on the previous slide, the mass spectrometer measures mass-to-charge ratio. In this example, verapamil accepts a proton to form the M plus H plus ion. So the mass-to-charge ratio is calculated by taking the monoisotopic mass of verapamil and adding the mass of a proton, then dividing by the charge. The result here is that verapamil has a mass-to-charge ratio of 455.2905 Daltons. The Dalton is the unit used in mass spectrometry, named after John Dalton. He was the scientist who, in 1803, first defined the weight of atoms. I just used the term monoisotopic mass. So before we go on, we should discuss some other definitions of mass. When we talk about mass in mass spectrometry, we often confuse different definitions of mass or use them interchangeably. So let's define some terms now. First, nominal mass. Nominal mass would be the mass we might calculate on the back of an envelope or on a scratch pad for a quick calculation, where hydrogen has a mass of 1, carbon 12, oxygen 16, and so on. Average mass is the mass calculated using the relative average isotopic mass of each element. The molecular weight listed on reagent bottles is an average molecular weight. The monoisotopic mass of a species uses the exact mass of the most abundant isotope, with the scale being set by the most common isotope of carbon, carbon 12. It is this mass that is typically measured by mass spectrometry systems. One other term it's useful to keep in mind is isotopic abundance. Isotopic abundance is how much of the naturally occurring distribution of a given element is due to a particular isotope. In mass spectrometry, we observe isotopic peaks in our spectra. So for carbon-based species, we would see isotopic peaks based on carbon-12 and peaks where some carbon-12 has been replaced by carbon-13. These definitions are very important when confirming or identifying a compound during your mass spectrometric analysis to ensure a correct result. Now that we've introduced mass spectrometry and defined some terms, let's look at the basic building blocks of a mass spectrometer. An MS system can be broken down into several parts. First, the sample introduction mechanism. Then, the mass spectrometer itself, which consists of the ion source, where the analytes are converted into gas phase ions, the mass analyzer, where ions are separated by their mass-to-charge ratio, and the detector, where the different mass-to-charge ratio ions are detected individually. Finally, there is the data system, which acquires the data from the mass spectrometer, processes it, and generates results. We will be focused on the ion source in this webinar, and part 2 of the series will look at mass analyzers and detectors. The ion source is where our analyte species are converted from neutral molecules into ions. This process is critical to ensuring that the species present in your sample are detected. Depending on the type of mass spectrometer being used, the whole MS detection system may be under vacuum from the ion source to the detector, or the ion source may be running at atmospheric pressure, and then the ions created will be introduced into the vacuum region of the mass spectrometer after the source via a sampling orifice or sampling cone. We will discuss both types of ion sources, both vacuum and atmospheric pressure, in this presentation. As well as discussing how ions are formed, we need to think about how we would choose an ionization technique suitable for our analytes of interest. As an example, naphthalene on the left and buspirone on the right are two very different molecules. How would we decide on an ionization technique for each molecule? What factors might be important in making that decision? When choosing a technique, a number of sample properties are very important. Analyte chemistry and the phase in which the sample enters the ion source can dictate which ionization technique is the most appropriate. The polarity of the species and the molecular weight of the compound can also be critical in choosing an appropriate ionization technique. The molecular weight may play a role as well, as large molecules may be fragmented if certain ionization techniques are used. This chart shows the usable mass and polarity ranges for several ionization techniques we will be discussing today. For example, if your analyte is very small and non-polar, there are a number of techniques that may work and several other factors would need to be considered. If your analyte is very large and very polar, there may be only one technique available to you. This chart is by no means exhaustive, and we will consider techniques not shown on this chart as well during today's webinar. So let's start to look at those techniques now. As mentioned earlier, we will be looking at techniques that work under vacuum, then moving on to techniques that run at atmospheric pressure, and we'll finish up with looking at some techniques known as direct MS. We'll start with ionization techniques that take place under vacuum. These are some of the earliest techniques used for ionization in mass spectrometry. In these techniques, the sample is introduced into a chamber or ion source under vacuum and is ionized there before entry into the mass spectrometer. Typically, samples are in the gas phase, though some systems allow for the introduction of solid samples into the ion source with a specialized probe. These techniques have commonly been used with gas chromatography. The first of these techniques is Electron Impact Ionization, or EI. EI creates ions by the displacement of an electron from the molecule by an electron beam. This is a very energetic process and produces fragmentation of analytes in nearly all cases. Molecular ions created by EI, if they are present at all, are radical ions. To move the created ions from the source into the mass spectrometer, a voltage is applied to a plate on the left-hand side of the source in our diagram called the ion repeller, which pushes the ions into the mass analyzer. In chemical ionization, or CI, a reagent gas, commonly methane, ammonia, or isobutane, fills the ion source. This reagent gas is ionized and interacts with the sample to produce ions. The ions then exit the source region into the mass spectrometer. The exit aperture in the CI source is much smaller than in the EI source to help contain that reagent gas, but it still allows ions to enter the mass analyzer. Here is a comparison of the steps in the ionization process for EI and CI. In CI, you can see the reagent gas R is ionized as a first step. This reagent gas then interacts with the molecule of interest to form M plus H plus in Ci rather than the radical cation M plus dot as seen in EI. The difference in ionization between EI and Ci also leads to very different mass spectra being observed. In EI, the molecular ion signal is small and a large amount of fragmentation is observed due to the energetic nature of the ionization. In Ci, the ionization is softer and leads to much more of the monoisotopic ion being observed as the protonated ion MH plus in the spectrum shown. In many cases, systems with ionization under vacuum can switch between EI or Ci ionization as needed by the analysis being run. In contrast to vacuum ionization, many modern ionization techniques occur at atmospheric pressure. This difference has led to a huge leap forward in the types of molecules that could be analyzed by mass spectrometry. Ionization at atmospheric pressure requires that the source must not only create ions, but any solvent associated with them must be removed before they enter the mass spectrometer. This process is known as desolvation. This process can involve the use of gas and heating of the ion source to ensure the desolvation process is efficient, and it is critical to ensuring good sensitivity for the mass spectrometer. Atmospheric pressure GC, or APGC, bridges the gap between traditional GC ionization techniques under vacuum, like EI and Ci, and atmospheric pressure techniques used with liquid chromatography. APGC uses an atmospheric pressure source at the end of a standard GC system. The gas flow from the GC mixes with a makeup gas which interacts with a corona discharge. This discharge ionizes the species passing it, and the ions generated then enter the mass spectrometer. APGC maintains benefits of traditional GC methods, but also has advantages such as being able to use faster gas flow rates in the GC, and the spectra have less fragmentation due to softer ionization. There are two primary mechanisms of ionization in APGC. The first is a charge transfer mechanism initiated by corona discharge ionization of the nitrogen in the source to generate radical cations of nitrogen. These radical cations then undergo charge transfer with analyte molecules to generate radical cations of those analytes. This form of ionization is particularly useful for the analysis of non-polar compounds. Alternatively, ionization can take place through proton transfer reactions. This example shows the proton source as water, but methanol or other solvents can be used. As seen here, a radical cation of water is created, and this undergoes proton transfer with the analyte to form the M plus H plus ion. It is possible to select between proton transfer and charge transfer in APGC by altering the conditions in the ion source depending on the chemistry of the target analytes. The spectra obtained via APGC show much less fragmentation than traditional GC ionization. In the example shown here, dibutyl sebacate has been ionized by EI on the left and by APGC on the right. The APGC spectrum does show some fragmentation, but we also observe a significant amount of the monoisotopic ion at mass to charge 315.25 Daltons. We'll now move away from ionization techniques associated with gas chromatography and move on to look at techniques more commonly used with LC-based analyses. Electrospray ionization, or ESI, is one of the most commonly used techniques in modern mass spectrometry. Professor John Fenn demonstrated its applicability to mass spectrometry, for which he won the Nobel Prize in 2002. Electrospray is a soft ionization technique. It ionizes a wide range of analytes and works best for compounds that are slightly polar to highly polar in nature. In addition, electrospray ionization can ionize compounds over a wide mass range. These properties help to explain why ESI is one of the most popular ionization techniques being used in mass spectrometry today. The buspirone example we saw earlier in the webinar is readily ionized by ESI as it is a polar molecule. Electrospray ionization involves applying a voltage, typically in the low kilovolt range, to a conductive capillary. This applied voltage induces the liquid flowing through it to produce an electrically charged aerosol spray upon exiting. The resulting spray then gives rise to ions contained in the aerosol droplets. The ions desolvate by interaction with heat and gases in the ion source. The ions then make their way into the mass spectrometer. A positive ion is produced in electrospray when a proton, shown in red, or an adduct is added to a molecule. Most commonly, that is a proton, but other adducts can be formed with ammonium ions or sodium ions, for example. A negative electrospray ion is produced when a proton is removed from a molecule, as shown in the bottom of the slide. Once the charged droplet is sprayed from the ESI probe, the droplets must desolvate to ensure the ions can be analyzed in the mass spectrometer. The cartoon shown here shows two possible mechanisms for how this could occur: the ion desorption mechanism or the charged residue mechanism. In the ion desorption mechanism, the droplet loses liquid by interaction with gas and heat in the source until a charged ion is released from the droplet. In the charged residue model, the droplet shrinks until charge is transferred to the molecule, creating the ion. ESI is widely used because it ionizes a wide range of analyte types. It works best for compounds that are slightly polar to highly polar in nature, and it can ionize compounds over a wide mass range from small molecules to large proteins. Some examples of species that ionize in positive electrospray and negative electrospray are shown on the slide here. While the examples we've seen show the addition of one proton to a molecule, one of the many benefits of ESI is that some compounds can accept more than one proton during ionization and become multiply charged. This allows the master charge of larger molecules to fall into the working range of many mass spectrometers, allowing analysts to interrogate very large molecules. As molecules become larger in mass, they can form a population of charge states at the same time under the same conditions. Many proteins will form such charge state distributions, similar to that seen here on the slide for horse-heart myoglobin. Here, we see a distribution of charge states from +24 up to +9, and the charge state for each of the species seen is shown in red on the spectrum. Another very common atmospheric pressure ionization technique is APCI, or atmospheric pressure chemical ionization. Ionization in APCI is achieved by passing the sample in solution through a fused silica capillary. As it emerges from the capillary, it is nebulized by a flow of nitrogen gas into a heated tube where it is vaporized. Ionization is produced by applying a high voltage to a corona pin at the end of the heated tube. The corona discharge around the pin causes solvent molecules to be ionized, forming a reagent gas plasma. Sample molecules passing through that plasma are ionized by proton transfer to form M plus H plus in positive mode or M minus H minus ions in negative mode. These ions then enter the mass spectrometer. Certain types of molecules can pick up or lose an electron and become either a free radical cation or a free radical anion in positive or negative APCI respectively. Here's the chart with different ionization types we saw earlier in today's webinar. APCI works well for low molecular weight species, typically less than 1,000 Daltons. Unlike ESI, APCI generates singly charged species, but fragmentation can occur easily in the source due to the energetic nature of APCI. Unlike electrospray, where polar mobile phases work well, non-polar solvents can be used with APCI. This allows users to run normal phase chromatography if required. In this table, we compare ESI and APCI. ESI and APCI are often run on the same mass spectrometer since much of the source is the same for both. This ability means that one mass spectrometer will allow a user to analyze a wide range of compounds regardless of the properties of the analytes in question. The main differences are these. In electrospray, ionization occurs in solution before nebulization, and in APCI, it occurs after nebulization. In APCI, the probe is heated, and in ESI, it is not. The more polar a compound is, the more likely it is to ionize in ESI. And the more thermally labile a compound is, the more likely ESI is a more suitable technique. All of these factors should be considered along with the factors discussed earlier in the webinar when choosing between these techniques. A technique with similar properties to both ESI and APCI is Unispray, a novel ionization technique developed by Waters. A liquid stream is converted into a nebulized spray and directed at a stainless steel target held at high voltage. Interaction with the target creates a fine spray of charged droplets. The resulting ions and charged droplets are sampled by the first vacuum stage of the mass spectrometer via a sampling cone. As you can see in the bar chart to the right, Unispray can provide greater ionization efficiency for some compounds over other techniques, giving analysts another option when looking at atmospheric pressure ionization. But what techniques are available to an analyst if their compound is very nonpolar, where electrospray or APCI may not work? One option is APPI, or atmospheric pressure photoionization. This is a complementary technique that does not use charge to initiate the ionization. Direct photoionization can occur when UV light is directed onto the molecule. This extracts an electron from the molecule to leave a free radical cation, as seen here. Alternatively, ionization can take place through a proton transfer reaction with a solvent that has been radicalized by the UV light. Useful solvents in this technique can be solvents such as water or methanol. Here is a schematic of the APPI source. Molecules enter from the top via a probe and are sprayed into the ion source. The UV lamp is on the right-hand side. It shines photons onto the sample molecules to create ions. Once ionization has occurred, ions are pushed towards the mass spectrometer using a repeller electrode and enter the mass spectrometer via the sampling cone. APPI can provide functionality and ionization not available via other atmospheric techniques such as APCI or ESI. It works well with polar molecules and is sensitive for compounds with a high proton affinity. It can also ionize very non-polar molecules such as the naphthalene we saw at the beginning of this webinar, and can ionize in both positive and negative modes. So far, we've examined techniques that can be used with chromatography, such as GC or LC, but are there techniques where chromatography is not needed? Direct MS ionization techniques encompass a group of techniques where there is no chromatographic separation. Direct MS analysis was common in the early days of mass spectrometry, where probes containing solids were inserted into the ion source and ionized. But with new techniques, such as the ones we will examine today, direct MS analysis has undergone a resurgence in the last few years. These techniques are fast and in many cases require minimal sample preparation, with the exception being MALDI. They can be less useful for complex mixtures and matrices, but do allow a whole range of compounds to be analyzed without an associated separation technique. Quantitation with these methods can be more difficult than those associated with chromatography. And the four techniques we will look at today will be ASAP, DART, DESI, and MALDI. The Atmospheric Solids Analysis Probe, or ASAP, was introduced by Charles McEwen, currently a professor at the University of the Sciences in Philadelphia. ASAP is a useful tool for the rapid, direct analysis of solid and liquid samples that are volatile or semi-volatile in nature. This technique harkens back to the early days of mass spectrometry, but occurs at atmospheric pressure rather than under vacuum. The ASAP technique utilizes the heated nitrogen desolvation gas to vaporize the sample and a corona discharge for the sample ionization. This technique is fast and sample runs are typically less than 1 minute in duration, and it also requires no sample preparation or chromatography. Direct analysis in real time, or DART, is a desorption APCI-based technique where heated ionized gas is directed at a target between the DART interface and the mass spectrometer. It can ionize molecules from the surface of that target, and that target can be a solid, a liquid, or a vapor. This ability makes it applicable to a broad range of applications. An electrical discharge in the source creates a plasma. This plasma contains ions, electrons, and excited neutral atoms and molecules. Electrostatic lenses in the DART source remove the ions and electrons, leaving only long-lived excited atoms and molecules known as metastables. A heated gas containing those metastables exits the source and heads towards the inlet of the mass spectrometer. The sample of interest is placed in that region for analysis. Ions are formed by interaction of the metastables with the sample. The heated gas also assists in desorption of the molecules from the sample target. The speed of generating mass spectra with minimal or no sample preparation makes DART an ideal tool when rapid analyses are necessary. Desorption Electrospray ionization, or DESI, was invented by Zoltan Takat in Graham Cooks's group at Purdue University in 2004. It employs a fast-moving charged solvent stream at an angle relative to the sample surface to extract analytes from the surface and propel the secondary ions towards the mass analyzer via a transfer capillary. The solvent stream is typically a combination of methanol and water, sometimes paired with a low percentage of acetic acid so analytes can be ionized. Ionization occurs due to the desorption of material from the solid to the gas phase at atmospheric pressure and interaction with the solvent. Analytes such as metabolites, lipids, or sugars work well as analytes in DESI systems. Matrix-assisted laser desorption ionization, or MALDI, uses a laser to create ions. A mixture of laser-absorbing matrix in the sample are coated onto a target plate with the matrix in excess in the mixture to ensure good ionization. The target plate is then placed in the instrument source, typically under vacuum, and a pulsed laser irradiates the target plate, triggering ablation and desorption of the sample and matrix material. The analyte molecules are ionized by being protonated or deprotonated in the hot plume of ablated gases, and then they can be accelerated into the mass spectrometer. MALDI has been applied to the analysis of biomolecules such as DNA, proteins, peptides, and sugars, and large organic molecules such as polymers, dendrimers, and other macromolecules. These molecules tend to be fragile and fragment when ionized by other ionization methods. It is similar in character to electrospray ionization in that both techniques are relatively soft ways of obtaining ions of large molecules in the gas phase, though MALDI typically produces far fewer multiply charged ions. Spectra from both electrospray and MALDI also show low fragmentation. So to summarize, there are a range of ionization techniques available in modern mass spectrometry, and we've looked at a number of those ionization techniques in this section of the webinar. It's also important to remember that the choice of ionization technique is related to the type of sample and the characteristics of the analytes of interest, and all of this should be thought about when selecting an ionization technique. As well as the ionization technique being used, the design of the ion source can be critical to the functionality of the mass spectrometer. We've touched on this topic a little when discussing ionization techniques, but let's look at it in more detail. In sources where the ionization occurs under vacuum, such as EI, shown here, the ions only need to be moved from the source into the mass analyzer as they are already under vacuum. Mass spectrometric detection takes place at pressures much lower than atmospheric pressure. So in atmospheric pressure sources, the ions must move from the atmospheric pressure region through some sort of orifice into the vacuum region of the mass spectrometer. In this picture, there are two designs for an ion source. In order to help move ions into the vacuum region, voltages are applied to the sampling orifice, and there is also some pull from the vacuum through the orifice as well. The most straightforward design of a source would be to place the spray directly in line with the sampling orifice. The orifice keeps the mass spectrometer under vacuum while still allowing ions to enter the vacuum region. Having the spray directly onto the sampling orifice of the mass spectrometer may affect the cleanliness of the orifice, and with particularly complex or dirty matrices, the orifice would require constant cleaning to keep up the instrument sensitivity, and in a worst case, could become blocked. Many vendors use an off-axis design, as shown in the bottom, to try and minimize the amount of non-ionic species introduced into the mass spectrometer. Doing this helps to minimize instrument downtime that might be required for cleaning the ion source. Having the spray off-axis from the orifice allows ions to enter the mass spectrometer using the voltages applied to the source and the vacuum of the mass spectrometer, but much of the rest of the matrix, which is not charged, does not hit the orifice or enter the mass spectrometer, which means both better uptime for the system and improved signal-to-noise. So what happens at the sampling orifice of the mass spectrometer? In the design shown here, there are two sampling cones, the inner and outer cone. The inner cone has a hole to allow ions to enter the mass spectrometer. The gap between the inner and outer cones in this design allows a countercurrent gas to pass into the source. This helps with ion desolvation and to keep non-ionic species away from the orifice, helping to keep the orifice clean. This allows more samples to be run on the mass spectrometer before cleaning the orifice is necessary. Once through the sampling orifice, some instrument designs use a tube after the initial orifice to assist with further desolvation and to transfer ions into the vacuum region of the mass spectrometer. One downside of this design is that the tube could become blocked with non-ionic species from the sample and may require either cleaning or replacement. Here is a diagram of the Waters Z-spray source design. This is the standard design for all Waters atmospheric pressure ionization sources. The ion beam is sprayed from the probe, and the ionic species in blue make two turns due to the voltages applied to the orifices in the source and the vacuum present. The first voltage occurs as they pass from the atmospheric pressure region in white into the first vacuum region of the mass spectrometer in light blue. And the second occurs as they move into the next vacuum stage of the mass spectrometer in dark blue. The non-ionic species are not affected by the voltages in the source and do not turn. Some non-ionic species may be sucked through the first sampling orifice by the lower pressures inside the mass spectrometer. While the first turn deals with the majority of the non-ionic species, the second turn ensures even less of them enter the system. This affects both the cleanliness and sensitivity of the mass spectrometer. Our ions are now in the mass spectrometer and move into the mass analyzer. The different types of mass analyzer will be covered in part 2 of this webinar series. In conclusion, there are a number of ionization options available today, and the technique chosen is closely related to both the application to be run and the compounds to be analyzed. The other thing that must be considered is the geometry of the source, which can have a large effect on the sensitivity of the mass spectrometer. In our next webinar, we will be discussing the heart of the MS system, the mass analyzer, and we'll also discuss the types of detection available.